In today's class, we are going to start with the topic absorption by roots. So in this, first we'll deal with the type of membranes that we see, and we'll also discuss processes that will come as we go through the topics. So first we are going to see what a freely permeable membrane is. And before that, let's see what solute and solvent look like. Solute are big size particles, whereas solvent are small size particles. An example being water, and example of solute is say sugar. So if we mix sugar and water, so water becomes the solvent and sugar dissolves in water, hence is the solute. Some basic differences between solute and solvent. So solute generally is smaller in quantity. Solvent generally is larger in quantity. Solute generally loses its identity, whereas solvents generally retains its identity. Think of sugar and water. When we mix sugar and water, sugar initially it was in solid state, but as we mix it in water, what happens is sugar dissolves in water, and at the end what we see is a solution. So sugar initially solid converts into liquid, hence it loses its identity, is a solute, and we generally take sugar in smaller amount as compared to that of water, and water generally in larger quantity, and it retains its identity. That means it was initially liquid and at the end also it remains liquid. So it retains its identity. So solute are larger sized particles, as we can see over here, and solvent are smaller sized particles. So this is the basic difference between solute and solvent. Okay, so freely permeable membrane, how does it look like? Freely permeable membrane looks somewhat like this, where it will allow the solute as well as solvent to pass. That is also why it's called freely permeable membrane, because solute can also pass through this and solvent can also pass through this. Okay. So since this is a free permeable membrane, as you can see, this size is big enough for the solute to pass as well as for the solvent to pass. So solute will pass from here to here and solvent moves from here to here. Now why does solute pass from left to right and solvent pass from right to left? Let's discuss the reason behind it. This is a high concentrated solution. Whenever the concentration of solute here on comparison, solute present here is more and solute present here is less. So this is called a higher concentrated solution, or basically we can say this as a concentrated solution. And this we call as a dilute solution. Okay, so over here solute can pass, solvent can also pass. So why does solute pass from left to right and solvent from right to left? This is because of a process known as diffusion. So what diffusion is? It is the free movement of molecules of a substance from a region of their higher concentration to a region of their lower concentration. When the two are in direct contact, so let's understand this with an example. Say we have a beaker and we have a water filled in this beaker. So if we drop a small color, what happens is this color will start diffusing, that is, it will start making the water color. So first it will distribute itself in this portion. Then slowly what will happen is that this color will start spreading farther and farther. So this is now color already, as we have seen. This portion is color. Now this will move further to color the other region as well. And slowly we will see that the whole beaker of water becomes colored. So this process is called diffusion, where this color was very highly concentrated in this region, and over here we only had water, the solvent. So from a region of its higher concentration, it started moving towards the region of its lower concentration as the two were in direct contact. So this starts moving, and slowly it will diffuse all over the water, and ultimately we will get a colored water which is evenly colored throughout the solution. So this process is called diffusion. See, this is potassium permanganate. Potassium permanganate is purple in color, and slowly it will make the whole water appear purple. example of freely permeable membrane. You can take as paper is a good example. Whenever you put some water above paper, any solution above paper, you'll see that below the paper the water will start dripping and the solution will remain intact. And you can also take perforated plastic. Perforated basically means containing holes. So if the plastic will contain holes, what will happen is the whole solution will pass through the plastic. So these are some examples of freely permeable membrane. Next, the second type of membrane we are going to talk about is the impermeable membrane. So impermeable membrane will not allow the solvent to pass, will not allow the solute also to pass. Let's see how impermeable membrane looks like. So impermeable membrane will look something like this, where there will be no space for any solute or solvent to pass. So though there is concentration difference, or one is more concentrated solution and other is a dilute solution, still we see that there is no transfer of molecules, there will be no movement of molecules of any substance, because this membrane does not allow any transfer of molecules. So this is an example. This is your impermeable membrane. And example would be, say, plastic. You cannot pass any water through plastic. You put any solution and take a plastic paper, put the solution above the plastic, you'll see that nothing comes out of the plastic from below. So plastic is an impermeable membrane. So next and the most important membrane that we are going to deal with is the semi-permeable membrane. So semi-permeable membrane looks something like this. Where So as we can see, the spaces between this membrane is such that we can have water molecules passing through them or the solvent molecules passing through them, but the solute molecules will not fit into this. That is why it does not allow the solute molecules to pass through it, whereas the solvent can pass through this. This type of membrane is called a semi-permeable membrane, and this process we learn is osmosis. So it is the movement of water molecules from a region of their higher concentration So where are water molecules highly concentrated? In this solution, and this solution is the dilute solution, and the water molecules, they are less concentrated comparatively in this solution, and this is our concentrated solution. So water molecules move from a region of their higher concentration, of their means over here, that is our dilute solution, to a region of their lower concentration. So water is less concentrated in this region, and this is the concentrated solution. So the movement of water molecules from a region of their higher concentration to a region of their lower concentration, and what are they passing through? Through a semi-permeable membrane. This is called osmosis. Now, suppose if this were to be the cell, what would happen is the outside concentration is higher and the inside concentration is lower. That inside is dilute and outside is concentrated solution. So from the cell, water would go outside, and if water would go outside of a cell, we call that process as exoosmosis. And if this were to be the cell, then water would have come inside. This process is known as endoosmosis. So we define these two terms, endoosmosis, where endo means inward. So the movement of water molecules inside the cell, where we are taking this as the cell, we are considering this as the cell. So water molecules are moving inside the cell. So the movement of water molecules inside the cell, when the surrounding region is concentrated, is called endoosmosis. This tends to swell up the cell. And exoosmosis, exo means outwards. So if this were to be the cell, what would happen if water would move outwards? So the movement of water molecules outside the cell, when in this case, in the endoosmosis, the movement of water molecules was inside the cell when the surrounding region was dilute. And in exoosmosis, the movement of water molecules is outside the cell. This is we are considering as the cell. So the movement of water molecules is outside the cell when the surrounding region is concentrated. Is called exoosmosis. You can also try an activity at home. You can take small raisins, you can put that in water, you'll see that the raisins will swell up. And if you put the same raisins in a sugar syrup, say, it will shrink further. So that is a very good example of endoosmosis and exoosmosis. And exoosmosis tends to shrink the cell. So next we are going to discuss what tonicity is. So tonicity is the relative concentration of the solution that determine the direction and extent of diffusion. Also note that osmosis is diffusion itself. There is no difference of a diffusion. So what happens is osmosis is the diffusion of water molecules. Here the diffusion does not take place of any substance. The diffusion is specified for the water molecules. So osmosis also is diffusion, where diffusion is taking place of water molecules from a region of their higher concentration to a region of their lower concentration, and with spatial importance to the semi-permeable membrane, where it needs to be passing through a semi-permeable membrane. Then we call it as osmosis. So tonicity is the relative concentration of solution that determine the direction and extent of diffusion. The solutions based on above can be of three types. Number one is isotonic solution, where iso means same and tonus means tension, concentration. So isotonic is when the relative concentration of water molecules and the solute on either side of the cell membrane is the same. So when the water molecules and the solute, the concentration of them is same on both the sides, then we say that it is isotonic. And over here, when kept through a semi-permeable membrane, this transfer of water molecules will take place only till when both the solutions become isotonic, where that is when they have the relative concentration same. Once it becomes isotonic, there will be no osmosis taking place. Because as you can see, osmosis is a movement of water molecules from dilute solution to concentrated solution. Once their concentration becomes same, that is both have the same concentration, so there will be no dilute or concentrated solution. As there will be no dilute or concentrated solution, there will be no movement of water molecules. So there will be no osmosis in case of isotonic solution. Second one is the hypotonic solution. Hypo means less or lower. So in this condition, the solution outside the cell has a lower solute concentration than the fluids inside. So here endoosmosis will take place. Solution outside the cell has lower solute concentration. So which one is less concentrated? This one. So this is the dilute solution. So solution outside is the lower concentrated solution. So think of this as the cell and the solution outside is the dilute solution. So what happens is water starts coming inside in this case. So hence endoosmosis takes place. So hypotonic solution, whenever placed in a hypotonic solution, the cell by endoosmosis will swell up. And third is the hypertonic solution, where hyper means higher. In this condition, the solution outside the cell has a higher concentration of solute. Then the fluids inside the cell. So exoosmosis takes place. Okay. Over here in hypertonic solution, what is happening is the fluid outside has a higher concentration of solute. So higher concentration of solute is over here. So this is the outside portion and this is our cell. So when the fluid outside have a higher concentration, what happens is water starts going out of the cell. As the water starts going out of the cell, what happens is exoosmosis takes place and we say that this solution is the hypertonic solution. So this is a hypertonic solution. Now we will discuss three definitions relating them. One is of turgidity. So turgidity is the state of the cell in which the cell wall is rigid and stretched out by an increase in the volume of vacuoles due to absorption of water. So when will a cell turn turgid is when a cell is placed in a hypotonic solution, because endoosmosis will take place. As endoosmosis takes place, it will increase the volume of the vacuoles, and as volume of vacuoles increase, it will push the cell towards the outside. The cell wall will become rigid and stretched out. Next definition is of plasmolysis. In plasmolysis, what happens is the contraction of the cytoplasm from the cell wall due to withdrawal of water when placed in a strong or say hypertonic solution. And last is the flaccidity. Flaccidity is the condition in which the cell contained is shrunken and the cell is no more what we call as tight. So what happens is, as we place a cell in a hypertonic solution, so exoosmosis will take place. A cell will lose water and the contents of it will shrink. So this shrunken condition is called a plasmolyzed cell or a flaccid cell, and when placed in a hypotonic solution, endoosmosis takes place and the cell increases in volume. The cell wall becomes rigid and stretched by an increase in the volume of the vacuoles. This condition is called turgidity and the cell is said to be turgid. With this, we come to the end of this lecture.